are going to talk about mitosis, but first I want to go a little bit over the actual cell cycle. It starts in G1 with cell growth, duplication of organelles, and protein synthesis, and then it moves on to the S phase where DNA is replicated and histones are synthesized, to G2 where final protein synthesis is completed, and then actual mitosis, which is going to be the focus of this video today. This includes prophase, metaphase, anaphase, and telophase, and these last two, anaphase and telophase, are actually a part of cytokinesis. The last G0 phase just depends on the type of cell that's being created. For stem cells, they stay in this period indefinitely, dividing and growing, whereas other cells, such as skeletal muscle cells, never actually enter. Moving on to the first phase of mitosis is interphase. And here we have our nucleus, where the DNA strands are loosely coiled and cannot be seen. And these yellow sun-looking things are the actual centrals and the centrosome. The next step is early prophase, which is right here. We have our nucleus with the coiled DNA, which can now be seen. Spindle fibers extend between each of the centrals, and pair smaller microtubules radiate into the cytoplasm, which is this yellow marker right here. Late prophase is next, and the DNA that was replicated in S phase that we spoke about earlier creates two copies of the chromosomes. Each copy is called a chromatid and is connected by a centromere, which is this light gray in the middle. Kintochores are protein-bound area of the centromeres that attach spindle fibers, forming the chromosomal microtubules, which you can see right here. Metaphase, down here at the bottom, the chromatids move towards the metaphase plate, which is a narrow central zone in the center. Metaphase actually ends once all of the chromatids are aligned. So that's what we've demonstrated here. The next step is anaphase, and the centromere of each chromatid pairs and splits, which you can see the different colors. And the sister chromatids divide. The two daughter chromosomes are pulled towards opposite ends of the cell along the chromosomal microtubules. Alright, in telophase, each new cell, which you can kind of see, prepares to return to interphase, and this right here, this is actually the cleavage frill that's getting ready for the actual division. The nuclear membrane reforms and the chromosomes uncoil, which is why we can no longer see them in the nucleus, and this actually is the marked end of mitosis. Cytokinesis, down here, is the actual division of the cytoplasm into two daughter cells, and it begins with the formation of the cleavage frill, which we discussed in telophase. Then this actually marks the end of cell division.